Government of Gujarat. She's also at the post of scientist C, and we are very fortunate to have her talk today from this platform. I would request without any delay to ma'am to start their lecture. Please, ma'am. With that, I would also like to invite Dr. Chirayu Desai, sir, to the National Conference on Environmental Microbiology and Regulatory Aspects. You're most welcome, sir. So very good afternoon, everyone. I still feel that people are sleepy. So I told Dr. Neepa madam that it is a very good time to take lecture because post lunch, I think students will be indulged in sleeping and I'll be, happy to wind up my job in no time. So jokes apart, am I audible enough to the last bench if they are interested in listening? Okay, thank you. So, thank you very much to Atmiya University and all the organizers, especially Dr. Rohan Pandya and Dr. Neepa Pandi for giving me this opportunity to come and interact with you. I still feel that my voice is not going proper, but if you are able to hear it, I am fine with that. So the topic that was given to me, or rather enforced on me by Dr. Rohan, was that you should speak on biosafety and biocontainment measures in the Indian landscape. So I said, okay, I'm not the expert. And he also knows that I'm not an expert to speak on this particular topic. But still, with whatever time that I've been allotted, I'll just try to give justice to the topic. So before I begin, and for those who are not sleeping, this session is not going to be a monologue, okay? So it will be a dialogue. So I will want students to also give me some of the ideas or some of the answers, and we'll see how the session goes forward. So what do you mean by safety? Can anybody tell me what is safety? Very simple. Safety. What is safety? Nobody? In Hindi, Gujarati. At least I know these three languages, Hindi, Gujarati and English. I mean, you can just say from wherever you are sitting if you are afraid of showing up your face. Safety? Salamati? Yes or no? So what is salamati? Are you safe in this auditorium? How can you say that? So suppose there is an earthquake, right? And the entire building collapses right now, right away. I don't wish to have an earthquake here, but let us assume if there is an earthquake. So do you still feel that we will be safe here? Okay. If there is a flood, suddenly, some rain pours and there is a flood, will we be safe in this auditorium? Yes. So you know, right? I mean, since the beginning, the first instinct of a newborn baby is to understand and identify the safe environment. And that's why, how one of the important instinct given to any living creature so that they can survive. So safety is very important. We are today going to see the biocontainment. What is containment? So I'm just trying to decode the words, so that you will have some perspective about what are we planning to understand today. What is containment? So according to me, all of you are willingly or forcefully contained in this seminar room. Is it right? I use both the words, either willingly or forcefully. So you are contained in this seminar room, isn't it? So you are just being put in a particular place for a particular time period. And now what is biocontainment? Any biological material or an agent that is put in a specific environment is biocontainment at the simplest. And what you see here is a beautiful picture of the work, India's second BSL-4 laboratory, which is coming up in Gandhinagar with the support from Government of Gujarat. And just a small correction, I don't work with Gujarat State Biotechnology Mission. I used to work till 2017. But from there onwards, I'm working with Gujarat Biotechnology Research Center, which is GBRC, which is one of the autonomous research center under Department of Science and Technology. So during this boring session, I would just try to cover, couple of things. What is biosafety? What is biocontainment? And what is biosecurity? Then what are the lab acquired infections, or what is in short called as LAI? Then what are the different levels of the biosafety do exist? And then international and national guidelines. And then a little bit story on how the BSL-4 project in the Gujarat is taking a shape for a future generation like you to take a use. Can I just have some water here? So, when we talk about biosafety and biosecurity, one of the important things to understand is a risk. And what I mean to show here in a slide over here is that risk is everywhere. Yes or no? Right now, we are taking some risk. Those who are even bunking this particular session, they are also taking risk, isn't it? So risk is everywhere. And wherever there is a risk, we try to put in the mitigation strategy. So as you can see here, the moment you step out, you can just flip yourself or maybe, fall on a road, or can happen anything with you. So there is a risk even in crossing a road. There is a risk in running a university as big as Atmiya University or any other university. There is a risk in running a company. Everything that you can imagine has got a risk. So what do? You stop doing it? No. You understand the risk, you identify it, and then just try to move on with that. So specifically, the audience like you, which is engaged day in and day out into all sorts of biological agents, whether they are microbes or viruses or fungi or any other, living organisms. I think some of you, I don't know whether you do animal studies or not. So whenever you deal with any sorts of biological agents, then there are some risks associated with it, and we need to understand those risks in order to mitigate it. So there are biological hazards. I'm sure when you will be doing, some experiments on DNA extractions, your faculty would have told you that mindful, whenever you use ETBR, it is a carcinogen. Yes or no? How many of you have heard that? So that means that whenever you are dealing with any of the organisms, it creates certain risks. For example, bacteria. So there are many pathogenic bacteria which are surrounding us. And those pathogens can enter knowingly or unknowingly or through some health hazards as well. So when you work with the pathogenic organism, there is a huge risk that those organisms can transmit to the person who is handling. So in order to create a biological environment, a safe place where you can perform various activities on those agents without harming yourself, is considered as a biosafety. Okay. So viruses, nobody needs an introduction on viruses like COVID, you have HIV. There are many, many pathogenic viruses. And today, once you go from here, you can just Google it on the Department of Biotechnology's website. that what are the list of different level of organisms, biosafety level organisms, and you will get a whole sorts of list. And then parasites. So in short, whenever you are talking about a biosecurity or a biosafety, you want to look at a physical security. Physical security means you cannot do any of the work anywhere. Can you perform isolation of E. coli at your house, in your kitchen, or here in this lab? No. So you need a specific lab, maybe a biosafety cabinet. If that is not available, at least a laminar airflow to work with. Even if that is not available, you would be working with the Bunsen burners, right? So you need some specified environment. Then personal security. How many times you people are scolded for not wearing aprons? Nobody? You're all wearing aprons? That means you are very good students. So personal protections like PPE. I'm sure all of you would know what is PPE. Personal protective equipments. So one of the PPE is your apron. Then you have a mask. Then you have an eye shield. Then you have gloves, so on and so forth. Then material control and accessibility. So suppose there is a live COVID in a bottle. Can you just open it here? That means you need to have an access. Who can have a live SARS-CoV-2? Who could have an anthrax in their bottle? So nobody could have it just like that. You need a specific measures and security too. So that means control and accountability, and then transport security. Have you seen, during the COVID time, I mean, there were big cartoons which were getting shipped with the swab samples. So those swab samples, whenever they are transmitted from one place to another place, there is a guideline of the ICMR that there has to be a triple packing inside the laminar airflow or a biosafety cabinet, and then surface sterilized, and then again packed it, and then it is shipped in a temperature controlled shipment. And most preferably, it should be logged also. So, and then information security. For example, there are people who are indulged into the activities of bioterrorism. So can you tell them that where you are storing all the high-risk pathogens? Yes or no? Can you tell them? So which means wherever you are handling these very high-risk organisms, you need to also have a security on information. What information can be given? What information cannot be given? So I'm sure you must be aware about NIV Pune, National Institute of Virology Pune, which is right now India's only BSL-4 laboratory. And it is housing a lot of, BSL-4 and BSL-3 viral pathogen repository for future uses and studies. So all those things are very important. But what are the challenges? So as I told you that you can encounter any of the biological substance either naturally or sometimes it is a risk when you work with them. Zoonotic diseases, for example, like COVID, okay, or leptospira. So any disease which is there either in the domestic animals or wild animals can infect human beings. So close to 60% of the pathogens which are causing infections in humans, they are zoonotic in nature. Then 75% of the emerging infectious diseases have an animal origin. So one or the other way, they were first originated in the animal. Microbiological laboratories, I am sure you would be doing your practicals. So your laboratories are also a different environment. If you do not work properly, you will be at the risk of getting yourself infected or your environment or your colleagues or your entire city, town, depending on the type of pathogen you deal with. And then these biohazards are very dangerous to mankind. And I'm sure many of the cases you would have been taught in your classes about. There was an anthrax covered letter which was shared in USA to one of the post mail, and then it was a classic example of bioterrorism. And then botulinum, which is a toxin coming out of Clostridium botulinum, which is being, put it into water bodies and stuff like that. So there are a lot of risks which are available. So one are natural ones, another one is acquired ones, which are laboratory acquired infections. So before moving forward, let us understand few of the definitions very right, because you're going to come across these words. So what are the biohazard? Any biological substance which has a potential health hazard to humans or surroundings, maybe some live organisms or the toxins out of it. Then what are the laboratory acquired infections? The name itself suggests that you are otherwise healthy, but you acquired those infections while not working appropriately in a particular laboratory environment. And then what are the biorisks and how do you do the assessment? So this is basically a process of evaluating the risks which arise out of biohazards, taking into account the adequacy of existing controls and deciding on to whether or not the biorisk is acceptable or not. And then the biosafety is a containment principle. So biosafety is nothing but you are just trying to contain organisms, technologies and practices that are implemented to prevent unintended exposure to the regulated materials and their accidental release. Till today, the entire mankind is debating whether COVID is a LAI or a naturally occurred infection. And there are no solid answers to it. And there are literally two groups of the scientists who are divided. Half of them say that it is a laboratory leaked virus infection which has created a pandemic, and many of the people, they say that it is transmitted from bat and pangolin to humans and stuff like that. And then finally the biosecurity, which is a security measures designed to prevent the loss, theft, misuse, diversion or intentional release of regulated materials and other related assets. So what are the LAIs when you work in laboratory? Most of your laboratory strains are domesticated, you could say, or they are non-pathogenic. But within E. coli also, there are many, many highly pathogenic strains which can create, infections to, if not entire city, at least to a society. So how do you contain them? So basically, LAIs occur into when the laboratory personnel contract infection within the lab. And what are the factors? Inadequate training is one of the most important, and that's why you are continuously hammered that whenever you are in lab, how you have to maintain your protocols. How will you do the apron? I mean, donning and doffing and all those stuff. Failure to follow SOP. You are told that you can open a plate only in the laminar airflow, but you open it anywhere. Stuff like that. And lack of PPE and other accidents. Sometimes if you follow everything, then also there are some accidents which can lead to this. Now, for example, if you look at the laboratory category, this is the data taken from one of the research paper where, the source of LAI. If you look at the research laboratory, how many LAIs have been reported? Almost 58% of the infections which are coming from the laboratory acquired infections are in research laboratory. Then diagnostic labs close to 17%. The biological product around 3.4%. Teaching laboratories also 2.7% LAIs have been reported, and many are unspecified. So what I'm trying to say that when you work with any deadly pathogen, does it mean that when there are risks, we should not work with them, or we should just, don't touch them, don't do research on them? No. How do you manage? You need some specific measures in place. But before that, how do you get exposed to this toxic substance? And mind me that today we are only discussing about biological hazards. There are a whole bunch of chemical hazards and other physical hazards which we are not discussing today. So inhalation is one of the common things. When you work with the cultures, for example, you work with a glass plate carrying an E. coli pathogenic culture which is isolated in a hospital, and you are moving along with it without putting it in a tray, and then you just drop it and then it gets released. Suppose it has a fungi, which means when you release it, not only the plate will break, but the fungal spores will go and spread into that surrounding environment. Ingestion. Sometimes, that's why you will see in the labs, mouth pipetting is forbidden. In most of the microbiology labs, you will see those codes. Why? Because accidentally, while pipetting out, you may suck those organisms into the mouth and ingest it. Contact with the mucous membrane and the skin. Suppose you are working with animal and you are developing a vaccine against leptospira. Leptospira is a human pathogen and you are doing a challenge study. So in challenge study, first you infect animal to see the efficacy of your previous vaccination. And rather than pricking mice, you prick yourself. And this is a very, very common mistake. I'm sure you can ask the people who have previously worked in those areas that this is a very, very common thing to happen. That means now the organism is within you. And I'm sure you would have seen a lot of Hollywood movies with, Tom Cruise and all, where Mission Impossible series is, some biological material, people just trying to protect it by, whatever ways and means. But those are not the part of biosafety protocols. Let me tell you, okay, it is all fiction. And then needle stick injuries. So, whenever you are accidentally exposed to any of the biological agent, what are the first things you should know? The first thing you should know is that the effect will vary individually. Every individual would have a different effect. Whether it is a lethal or it can only cause some infections. What is an ID50? Which means the number of organisms required to cause infection. What is its LD50? That means the amount of the agents which is required to kill at least the 50% exposed individuals. And what is the environmental stability? How does it get transmitted? Through water or through, aerosol or through air? Then what is the antibiotic resistance? What is the route of entry? What are the treatments or prophylaxis which are available? Whether it is endemic organism or exotic, or what is the host? So these are the parameters you need to consider for any LAI to understand what is going to be the consequences. And how do you do the risk assessment? So risk is what? The probability of an adverse effect occurring as a result on an exposure to hazard. And how do you do assessment? Evaluation of various parameters, as I told you, science, observation, experience, professional practice or SOP standards, and some common sense, which is not very common. And then risk assessment is what? Proactive rather than reactive. So you have to always predict the risk before it happens. It should never be the case that, jaise hoga to hoga dekha jayega types. You can't work in the high risk pathogens. What are the other ways to prevent these LAIs? Is by giving adequate training, providing engineering controls like, safety cabinets, ventilation systems, reduction of the exposure, and then administrative controls, for example, developing SOPs or enforcing good laboratory practices, also putting in, access control, who is allowed to go in that room for how much time, for what activity, etc., etc. So now, this is a very, very important slide. And if you want to move forward in my entire presentation, if at all you are interested, then you should be focusing on this slide, that how do you assess the risk and how many types of the risks are there when you are working with the biological agents. So typically, globally, all the scientists have divided that there are only 4 risk groups. Risk group 1, 2, 3, and 4 RG. What is risk group 1? That is no or low individual and community risk. For example, Bacillus subtilis. It has been given a GRAS status. What is GRAS? It is generally regarded as safe. You are eating a lot of probiotics whenever you visit a doctor, and when he prescribes you an antibiotic, along with that will be given lactobacillus and many other bacteria. So you are ingesting them otherwise also. Why? Because they are not known to cause any of the infections. Then risk group 2, which means you have a moderate individual risk and a very, very low community risk. So it can cause a disease, but unlikely to be a serious hazard, maybe limited to an individual or the individuals who are exposed. Then the risk group 3, which is a high individual risk but low community risk. So SARS-CoV-2, which is an causative agent of COVID, falls under risk group 3, which means if you are working with a live COVID, which means that you can have a high risk. And finally, risk group 4, which is a very high individual and very high community risk, specifically those pathogens for which neither you have vaccine nor you have treatment, and there are not. Are not means ability of one infected individual to infect other people is also very, very high. Can anybody in this room give me one example of risk group 4 organism? Like I said, COVID is group 3. Anybody? Any guess? Yes? HIV is not BSL-4, it is BSL-3 pathogen, risk group 3 pathogen. I mean, I know you know, I am asking from students. Ebola is the right answer. From India. Chalo, anybody? Which are the infections in India which are classified in risk group 4? And I'm sure when I will say the name, all of you would have heard it. Nipah virus. Have you heard Nipah infection in Kerala? So that is a risk group 4 organism. In Gujarat, which organism infection is present in Gujarat which classifies as risk group 4? CCHF, Crimean Congo hemorrhagic fever. Okay. And the cases have been very recently reported in Bhuj and the district around it, which is a tick-borne infection basically. So now you understood that based on their ability, of infection and its ability to cause the effect, after effect, there are 4 risk groups. But how do you manage the risk assessment? So in this matrix, you have to work. So this matrix says that from the level A, B, C, D, E, which means the likelihood of an event, whether it is almost certain, whether it is likely, possible, unlikely, or rare, which means A is definitely it's going to happen. E means that very rarely it can happen. And what is the impact? If that event happens, what is going to be the impact? So insignificant, minor, moderate, major, or catastrophic. So which means that insignificant, almost certain, insignificant event will fall into lower risk or higher risk. From the students, again I am repeating, almost certain infection with an insignificant impact will fall under lower risk or higher risk? Lower risk. Because it is almost certain that infection will happen, but it will only happen to an individual which will be easily cured out of that infection. But a rare event with a catastrophic effect on the society will be classified into what matrix? Higher risk. So that is how you develop a likelihood and then the consequences matrix. So as you can see that almost certain, insignificant is moderate, B likely is also moderate, and possible, unlikely, and rare is very, very low. Wherein here, on the scale of catastrophic, even the almost certain catastrophic event has the highest risk. But even if you will consider the E, which is a very rare event like a SARS-CoV-2 leaking out of Wuhan Institute of Virology, which was otherwise considered to be a very, very lower risk, also had a catastrophic effect very high because it impacted the entire world. All right. So now coming back to the main thing, when you want to deal with these organisms, we are all thankful that we are sitting here today in this auditorium because we could get vaccine on time. We could get vaccine on time. How many of you have seen the movie which is Vaccine War? How many of you have seen it? Only very few people. I would really advise you that your Netflix or other subscriptions you should be using on seeing a movie like this. So, in the vaccine war, those SARS-CoV-2 viruses were cultured in NIV Pune, handed over to Bharat Biotech so that they could grow it in liters, inactivate it, and create Covishield. Sorry, Covaxin. Covishield is an mRNA vaccine. And that's how we got vaccines. So you need to do research. But when you want to work on the deadliest organisms which are falling into the risk group 3 and higher, how you should be working, what should be the area specificities and all. I know this is a very, very boring talk. Believe me, even I never studied this. But if you want to continue your career in microbiology or biotechnology, then these are some of the very, very fundamental things you should be knowing. Otherwise, you will do some catastrophic event to the world, okay? And there are 4 risk groups. So to handle each of the risk group, there are 4 different biosafety practices known as BSL-1, BSL-2, BSL-3 and BSL-4. So this is a very basic level, like your practical laboratory, okay? Which is very good for the teaching and some sort of basic research. And there are good microbiological techniques, but there are no safety equipments required. So you just work with a Bunsen burner and streak some cultures, do gram staining, do some of the biochemical tests and just get over with it. Then BSL-2. Where do you need BSL-2? Primary health services, diagnostic and some level of research. What are the laboratory practices? So GMT, that is a good microbiological technique, plus you need protective clothing and the biohazard signs at an appropriate places. So one bench plus a biosafety cabinet and for potential erosis. Then comes the higher containment facilities like BSL-3. So in India, we have only around 70 to 80 functional BSL-3 laboratories. Okay. And those laboratories are either in public funded research institutes or mostly the vaccine manufacturing companies which are in the private. So these are special labs which have the BSL-2 plus some special clothing and controlled access and a directional airflow. Directional airflow means that when air is moving, it can only move in one direction. It cannot go reverse. So suppose another basic question. There is a room air which is there and then there is an airflow outside. If I open this door, under which circumstances the outside air will not enter this auditorium? What do you want here? You should have a negative pressure. Negative pressure means the pressure of the air is here low and outside is high. So whenever you will open it, the air will move out and then it will not come in. It is again very, very opposite to GMP. When you work in the companies, they follow good manufacturing practices. So the inside air is always going out, but nothing should come inside. But in the laboratories like BSL-3 and BSL-4, it is other way around. If entry is free, but exit is very, very strict. So, what are the biosafety levels? As I told you, I think we can skip this in the interest of time. These are some of the pictures I wanted to show you that how this high containment or maximum containment laboratories look like. So can you see these two people? What are they wearing? PPE. Okay, which level of PPE? Do you know that? Which level of PPE? Huh? Okay. Anyways, so these PPE are usually made up of a non-absorbent material so that anything from the outside cannot touch their body. Usually covered in a double mask system like the doctors used to go during COVID times in the hospital. You would have seen that. And what is this chamber where they are just, inserting their hand and doing some work? What is this? It's a type of biosafety cabinet, but there is a special terminology for this is called as isolators. So which means that inside, also there are some of the stringent environment of the air movement and things cannot move out. Now what is this? It looks more like a space station, but what is this? This is a typical BSL-4 laboratory, okay, where individual is wearing a positive pressure suit with a breathable air. So these red wires, they are connected to a chamber which is supplying a sterile air for breathing to the people who are wearing this specialized suit. And these are the positive pressure suits, which means when you are wearing them, you can work with one of the most deadliest viruses like Ebola, Nipah, CCHF, and etc. in this environments. Okay. So whenever you see a positive pressure suits, it is a BSL-4 environment. Now tell me, what is a biological safety cabinet? Have you seen it? Is it part of their curriculum? Biosafety cabinets? How it is different from laminar airflow? Can anybody tell me? Or whether they are same or different? That I know because I asked the question. So now tell me how it is different. How biosafety cabinets are different than laminar airflow? Just give it a good guess. Don't worry even if you give a wrong answer. Biosafety cabinets are specifically designed with a negative pressure so that the airs are, whenever they are circulating, the air cannot move out of that biosafety cabinet. Okay, that is one of the most thing. Where in the laminar airflow, the sterile air is moving out. So whenever you are working with the pathogenic organism, if you work in a laminar airflow, it is a catastrophic thing to do. For working on the pathogens, you must have worked only and only in the biosafety cabinets. And how many different classes are there? Class 1, Class 2A, A1, 2A2, Class 2, B1, B2, and Class 3. So Class 3 biosafety cabinets are those isolators as shown in the previous pictures. So mostly these are isolators where no air can move out. And in BSL-4, with a positive pressure suit, you cannot work in Class 3 cabinets. So most suitable is Class 2 B2 type when you want to work in BSL-4 environment. Okay. And as you can see that what are the pressure requirements, and as you can see that from Class 2A2, which is the basic biosafety cabinet to work in any microbiology laboratory, would have a negative pressure, all of them. Class 2A1 would have a positive pressure. And class 1, you can also work with a hard duct in an open rules. Now coming back to another boring topic out of the presentation is the guidelines. Suppose I don't follow any protocol, who cares? I mean, helmet is compulsory, but I never wear it, although it is for my own safety. Who cares? Right? But if you do that in the biological sciences and you want to work with the pathogen, first thing you will be harming is yourself, then your surrounding, and then the society at a risk. So there are different guidelines to adopt when you want to work with the high containment or the different biosafety level laboratories. So CWA, which is a laboratory biorisk management standard. Then there is a laboratory biosafety manual from the WHO. All these are available for free in PDF format in Google. And you can download it and just try to understand some of the terminologies. Specifically, those who want to work with the high-risk pathogens, this is going to have very, very valuable information. Then Canadian Biosafety Standards. CDC also has the BMBL guidelines, which is biosafety for microbiological and biomedical laboratories. And then Biorisk Management Laboratory Biosecurity Guidance, WHO. And biosafety biosecurity standards for managing biological risk in a veterinary lab and animal facility, which is a OIE guideline 2018. What are the guidelines available in India if you want to work with this type of organism? So the topmost act in the India is a EPA, what we call it as an Environmental Protection Act, which was of 1986. Then that is an Apex Act. Under the Act, there are rules, which is a Biosafety Rules 1989. And then the rules for manufacture, use, import, export, and the storage of hazardous microorganisms and GMO. And then the Biomedical Waste Management and Handling Rules 1998. So what are the biosafety rules basically? Department of Biotechnology, Government of India has released this and the notification, is from MOEF, Ministry of Environment and Forest, which talks about working on the genetically modified organisms as well as hazardous organisms and any activity which are associated with the manufacture, use, import, export, storage and research. I'm not going to read all these guidelines which are published by Department of Biotechnology. There is a very good portal where entire biosafety and biocontainment rules from different agencies like ICMR, WHO, everything is available. Some of the things that we want to understand here is that guidelines and handbook for the institutional biosafety committee. This is very, very important. Anybody who is working with a pathogen in the research laboratory, or if they are doing any sorts of genetic engineering, including some cloning experiments that you do anywhere, it requires a clearance from IBSC, which is Institutional Biosafety Committee. So how, when you want to work on a pathogen or a GMO, the rules specifically for GMO is an RDAC. So this is an apex committee which reviews the national and international level recommendations which are suitable for the appropriate safety and regulation in an Indian environment for recombinant research, use and application from time to time. So this only has an advisory role. If you come to approvals, then the apex committee that works under Ministry of Environment and Forest is GEAC, Genetic Engineering Appraisal Committee. So all of you are wearing cotton cloths which are manufactured in India. What is one striking thing that others may not know, but you being a biotech student should know, that they are all produced by BT cotton. And this BT cotton, which has a Bacillus thuringiensis gene, so that it prevents, an infection from a cotton bollworm and other stuff. For the cultivation of the BT cotton in India, the approval is given by this agency, GEAC. And we are very fortunate. Our director, Professor Chaitanya Joshi, is a member of the GEAC. Under GEAC, there is an RCGM, that is a Committee on Genetic Manipulation. So GEAC works under MOEF and it gives all the approval. Do you know recently India released another GM variety in agriculture, which is that BT mustard. BT mustard has been recently given a clearance for release, and it would be coming into the market after some of the containment trials are done. And then this RCGM, which is there in the Department of Biotechnology, which monitors the safety related aspects of the projects and activities. And then there is this IBSC, that is Institutional Biosafety Committee. And the enforcement is done at state level. If somebody is doing illegal genetic manipulation of plant, microbe or anything, then what are the powers given to the state government? The smallest one is a DLCC, which is a district level coordination committee, which is chaired by the collector of a particular district, and he has authority to destroy any of the GM material if encountered which is not approved by GEAC in any of the trial setup. And then there is an SBCT, which is a State Biotechnology Coordination Committee, which is chaired by not less than the Chief Minister of the state, involving the agriculture ministers, forest ministers. Science and technology was not into the picture, so now we are trying to push that DSC also has to be a part of SBCC. And last but not the least, these rules are very, very important for the students, which is a biomedical waste rule 1998. So whenever you go to hospital, you would see some color-coded dustbins are there. Do you think that they like it? Some red, yellow, black beans. They like those colors, or they are some specialized beans? Specialized beans, right? Very good. Do you have them in your labs? How many colors are there? Red, yellow, I spoke, and then blue is there or not? Okay, and then black is there or not there? Anyways, so when you talk about any waste, either it is a non-infectious or it is infectious. If it is a non-infectious, then you just simply classify it like a domestic waste, which is biodegradable and non-biodegradable. What your municipality calls it as bino kachro, suko kachro. Yes or no? And then in the infectious material, it is non-sharp and the sharp. So sharps are always disposed separately, preferably on site. If you have gone to a path lab, you would have seen, just after taking the blood, there is a destroyer, syringe destroyer, and they would just break it over there, and that the sharp part is collected and the syringe is thrown in a red color bag. Yes or no? And then non-sharps are solids and liquids, and again in the solid, there are two, one that can be incinerated and the other that cannot be incinerated. So what are the key rules? One of the rule is that wherever you are generating the waste, you have to segregate it, because you only know what is infected, what is not infected. It cannot be that I am generating waste, but somebody else does the classification. So biomedical waste shall not be mixed with the other waste. It should be segregated at the point of generation. And how to segregate it? For that, you need to refer to the schedule 2 and 3. If the container is transported from the premises, the container shall, apart from the label, prescribe also what are the schedules as per 3 and as per 4. And the untreated biomedical waste shall be transported only in the vehicles which are authorized for those purpose. In Gujarat, who does that authorization? GPCB, Gujarat Pollution Control Board. Okay. And no untreated biomedical waste shall be kept and stored beyond the point of 48 hours. So if you are storing it beyond 48 hours at a site, it is again a punishable offense against the biomedical waste rule. What are the categories? I am not going into detail, like human waste, animal waste, microbiology and biotechnology waste, sharp, discarded medicine, contaminated solid waste which are other than sharp, then the liquid waste, incineration ash, and the chemical waste. So there are 10 different categories under the schedule. And under the schedule 2, you would have these classification. So yellow is for plastic bag for category 1, 2, 3, and 6. Red is having, disinfected container or a plastic bag of category 3, 6, and 7. The blue, white, or the translucent ones are always in a puncture-proof container or a plastic bag for category 4 and 7. And the black would have category 5, 9, and 10. And how do you do the treatment? So it is given over here. So now I'll just take another 5 minutes to talk about one of the very, very interesting project of the state government, which is development of the second BSL-4 laboratory in Gujarat. First one is where? Pune. I told you, right? So NIT Pune is the only functional BSL-4 lab in India. And just imagine, this is the picture of the global BSL-4 laboratory networks. You can see they are heavily, placed in an European region, US region, and the parts of China, Japan, Australia, and even some in Africa. But the largest populous country like India, which has so many pathogens, we had only one BSL-4 lab which is working. Okay, and this is the classification. So if you just count that on per crore of a population, how many BSL-4 laboratories you have got? I mean, this is India at the rock bottom, right? And you know that our hygiene practices, the temperature which is most conducive for the growth of any microbes in the world, how much of the infections are there? So we just have one operational and we need many. So Switzerland has the highest number of labs per 1 crore population. And we have a lot of, risk group 4 pathogens, as I told you, SARS, which is SARS-1, okay? Foot and mouth disease, FMD, which is an animal disease, is also BSL-3 pathogen. Anthrax, avian influenza, H5N1, Nipah, Crimean Congo hemorrhagic fever, Canasur forest disease virus, which is today also active in parts of Karnataka, and any other high-risk group. So for investigation of any outbreak of an unknown region, you need to have this. Then Gujarat, as I told you, is endemic for CCHF, which is a BSL-4 pathogen. And then Nipah, we know that there have been several outbreaks. And to investigate this, this is the map of Gandhinagar, which is the capital of Gujarat. And this area that you see is the 14 acres of land which state government has given for developing a BSL-4 laboratory. And what are the salient features of these labs? You could use it for diagnostic, preventing, and therapeutic solutions. You can, this facility will also be shared with a competent, the word is very, very important, competent academia and industry to come and use it. It will also act as a national virus repository for the risk group 4 and risk group 3 pathogens. And it will give us even a capacity to deal with the agents of bioterrorism whenever there will be, some time. These are the modules. We are going to make one BSL-4 module which can deal on the human pathogens, testing and experimentation, series of BSL-3 laboratories for human pathogen, animal pathogen, and also an ISO 7 BSL-3 and BSL-2 laboratories. This is going to be the country's second lab with an outlay of 225 crore rupees. There are 12 modules, and this is how it will look like. So this is just a master plan. As you can see, this is an administrative building over here. And this area that you see is going to be the maximum containment facility. These are some of the utility area, and this is area kept for future expansion. If you just look at the block diagram, these are the BSL-4 laboratories. These laboratories are going to be BSL-2, including ISO 7 BSL-2, and these are series of BSL-3 laboratories. So even for entering, there's a so like when you want to do research in space, you need a specialized aptitude. Similarly, if you are willing to work in such high containment facilities, you need a very specialized aptitude. There are stringent protocols. When you move out, you are mandatorily taking 180 seconds, 3 different showers with 45 liters floating on one time. And when you work, that is for BSL-3. If you work in BSL-4, then your positive pressure suit has to undergo a chemical shower for disinfecting the entire surface. Then you remove that suit within the BSL-4 lab, and while doing your exit, you have to again come out of and shower out facilities. So shower out facilities means that you have to take a shower of 180 seconds 3 times each, and then you can make exit. So there are very stringent protocols for going in, moving out. You have to always work in a buddy system. That means an individual is not any day allowed to work alone in a high containment setups, which is BSL-3 and BSL-4. Always there has to be 2 people, which is called as buddy system. So the other person's job is just to observe that the person who is actually working with the pathogenic organism is not making any mistake accidentally also. So that is it, and thank you very much. I took a little longer than 1 hour. I hope that's okay. So I'm sure that there won't be any questions, so I'll step down. But if there are any, then you can raise your hand. Yes, ma'am. Please. I'm glad somebody has a question.
That was an excellent presentation, Madhvi.
Thank you, ma'am.
I have a question. Has there been any cases where the pathogen from category 2 has been now shifted to 3, or 3 has now been shifted to 4, like that? Like the in virus, like you have mutations and they become more.
So this was initially a part of BSL-3, which has now moved back to BSL-2. And again, these risk groups are very country specific. So you will have to keep in mind the criteria of endemism. So for example, foot and mouth disease virus, which is very much endemic to India, but it is not present in UK. So in UK it is considered as BSL-4, but in India we consider it as BSL-3. Thank you. And DBT every year updates the list. So now there is a 22 list which is available 2022, but every year we come up with.
Thank you so much, ma'am. I take the subject of biosafety and IPR. Now I know what things I had to incorporate for the students. So thank you. It was a material for me too. Thank you. A big round of applause for ma'am, please. I would request Dr. Neepa Pandey, madam, to felicitate ma'am. Thank you, ma'am. Moving on, we have now the next session from Dr. Chirayu Desai, sir, who is the PhD from SP University, who is now at Charosat University of Science and Technology, Charosat, Gujarat. His research areas include environmental biotechnology, and he has various projects funded from SERB, DBT, and he has also been funded for the National Mission of Clean Ganga and various projects sanctioned by GSBTN. We would be fortunate enough to listen to you, sir. Over to you.
So good afternoon, everyone. So this is the last lecture. So hopefully you have full josh and energy. Okay. So firstly, I am thankful to the organizers of EMRA 2024, especially Dr. Rohan Pandya, Dr. Nipa Pandi, Dr. Abhijit, and Dr. Raksha for inviting me here to share my with the younger generation. So today I will discuss our research on integrated bioelectrochemical systems and associated microbial communities for green energy generation and while treatment of hazardous industrial wastewater. So, which industry is much more relevant to Gujarat that you know of? Textile and dye industry. So we all are wearing colorful textile products, but it comes at a huge cost to the environment. So as you can see in these representative images, so image on the left would be much more familiar here, is that of Bhadar River in Jaipur, where around 80 lakh gallons of textile dyes containing wastewater is dumped on a daily basis by nearby sari printing and units and various textile printing units. Similar is the situation in Netaji Apparel Park in Tirupur in Tamil Nadu, where a water body is contaminated by the wastewaters which are dumped in the by nearby textile dyeing industries. Likewise is the situation in Sabarmati in Ahmedabad, where textile wastewater is mixed with the sewage and released through stormwater discharge systems into the Sabarmati River. So mainly the reason for this problem is the textile processes are water intensive. So processing of 1 ton of finished textile product would require 2 lakh to 4 lakh liters of fresh water, which is then converted into wastewater, which has resulted in global annual discharge of 280,000 tons of textile dyes into the water bodies across the globe. So why this wastewater is a concern is it is not only a serious threat to the aquatic ecosystems, but it is also carcinogenic, mutagenic, teratogenic to the human health. So the treatment of such wastewater is very challenging, mainly because it's a heterogeneous mixture of various concomitant xenobiotic pollutants like unfixed textile dyes, intermediates, surfactants, solvents, heavy metals, and auxiliary chemicals, which make them highly recalcitrant to biological degradation. So in any case, the dyes which are manufactured for their fastness and recalcitrance to photodegradation and biodegradation. Otherwise, you would lose the color on the fabrics. So that's why treatment of such wastewater is challenging. Moreover, the conventional treatment processes in the effluent treatment plant, like common effluent treatment plant, what is the most common effluent treatment process that is applied? Biological. Can anybody name? Yeah, so students, which biological wastewater is most common we apply in the effluent treatment plant? So madam answered that is activated sludge. Okay, so activated sludge, the conventional treatment process is not cost effective. We will require 0.5 kilowatt hour energy to treat 1 liter of wastewater. Okay, which accounts for almost half of the actual treatment cost. So this has led to generation of this situation where the untreated wastewaters are released into the surface water bodies because small scale industries cannot afford the treatment cost. So what if you devise an innovative solution or sustainable solution which can not only treat the wastewater but simultaneously recover electricity in form of bioelectricity? So this is possible using bioelectrochemical systems. So can you name any bioelectrochemical system, students? Microbial fuel cell. So what is the working of a typical microbial fuel cell? It's a conventional dual chamber microbial fuel cell which utilizes the chemical energy in the organic pollutants within the wastewater and converts it into the bioelectricity. So these microbial fuel cells are made up of 2 chambers. One is the anode chamber and a cathode chamber, separated by a proton exchange membrane. Okay, so the anode is maintained under anaerobic conditions and the cathode is maintained under aerobic conditions with aeration and a catholyte solution. So the inoculum of your bacteria, especially the electrochemically active bacteria, is inoculated along with the wastewater in the anode chamber, and these electrochemically active bacteria would form a biofilm on the surface of the anode. So these electrochemically active bacteria will then degrade the wastewater pollutants and generate protons and electrons. These protons will then migrate from anode to the cathode via the proton permissive membrane, which is mostly the Napier membrane that is used. And these H plus will then be utilized along with oxygen to accept the electron. So the electron that is produced by the microorganisms in the anode will be donated to the anode via various mechanisms, and these electrons will migrate through the external circuit and reach to the cathode, where this oxygen will accept this electron, forming water along with the proton, and this will generate a potential difference in the external circuit that will result in generation of bioelectricity. So now there are two mechanisms. Not all organisms will be electrochemically active. So there are two different mechanisms for electron transfer to the anode. So that includes direct electron transfer, in which there is a direct physical contact of the microorganism with the anode electrode, which forms pillins or extracellular membrane-bound cell organelles like cytochromes or pillin proteins or nanowires or filaments and appendages, through which the electron can directly be transferred from the microorganisms to the inert anode electrode. Whereas indirect mechanism involves redox mediators that will themselves get reduced and then oxidized and donate the electron to the anode electrode, such as quinones, flavins, and filaments. So the electrodes in the bioelectrochemical system, so this is the conventional bioelectrochemical system, that is a dual chamber MFC, are either made up of stainless steel or graphite. So either graphite or metal or carbon based or metal based materials. So this is the typical representation of a dual chamber MFC at the lab scale for degradation of reactive red 152 dye containing wastewater. So in this slide, we describe an mechanism of an electrochemically active bacteria in treatment of RR152 wastewater. So at 0 hour, you can see the dye is inoculated in the anode, and after 72 hours, the Nealia circulans DC10, that is a strain which is isolated, and this is a pure culture which achieves efficient decolorization by 94% decolorization and greater than 89% COD reduction with a maximum power density output of 166 milliwatt per meter square. So upon scraping the biofilm, so normally you study the biofilms through scanning electron micrographs, and if it is a consortium of bacterial electrochemically active bacteria, you would perform high throughput metagenomic sequencing. So in this case, we performed genome sequencing and we identified the mechanisms of Nealia circulans DC10, and we also identified the gene expression for genes that are involved in direct electron transfer. So the PIL-T and PIL-Z gene is what we identified are involved in the direct electron transfer to the anode electron. For dye decolorization, we identified an inducible azoreductase gene that will perform reductive cleavage of azodyes. So similarly, if we have an electroactive bacterium community or a consortium, then we follow a high throughput 16S rRNA gene sequencing or a high throughput metagenome sequencing. So in this case, we collect scrape the biofilms from the surface of the anode and then isolate the metagenomic DNA associated with the biofilms using an optimized method. And then if we are going for V3, V4 region, we'll follow this amplicon sequencing approach. Or if we are going for shotgun metagenomics, we construct the library and then sequence using the Illumina NextSeq platform, mostly by outsourcing, and then analyze the results or the sequence that we get through time to program. And also multiple metagenome analysis programs are used to identify the functional genes. So this is the methodology that is followed in all of our experiments for studying the microbial community associated with the bioelectrochemical treatment system. So the dual chamber MFC, even though it has been demonstrated in many studies, if you find, you will find multiple studies of a microbial fuel cell demonstrated for treatment of textile dye wastewater. However, one of the major limitations of this technology is the scale up. Because at lab scale, they give very good results, but when you scale up these technologies, they become less affordable, mainly because of higher cost of the proton exchange membrane. There is an epsilon membrane which is used as a separator in this system. So for in situ treatment, you require a technology which can be scaled up and applied at on-site. So constructed wetlands, on the other hand, offer several advantages like easy setup, lower maintenance cost, and long-term operation. And these systems are self-sustainable, so you need not require much maintenance if you have once established a wetland system. So what we came up is with a hypothesis that we can integrate a microbial fuel cell with a constructed wetland system, because constructed wetland has various natural stratified redox gradients. And for integration of an MFC, what you require the anode to be maintained in an anaerobic environment. So that can be possible if the anode is placed at the bottom of the wetland. And the cathode you need to maintain at an aerobic environment near the rhizosphere or at the air-water interface. So it can be placed near the rhizosphere. And you select plant species which are recalcitrant and that can degrade the azo dyes along with the rhizospheric organisms. Then you can develop a newer hybrid system that is called constructed wetland microbial fuel cell system. So initially, we tested this hypothesis by developing 3 identical systems like a constructed wetland microbial fuel cell system 1, which and 2, along with a constructed wetland system which does not have any efficiency of a constructed wetland microbial fuel cell upon integration of the electrode. Likewise, when we compared, as compared to the tap water fed CWMFC1, better efficiencies in terms of power density generation were obtained in CWMFC2. So there is 198.8 milliwatt per meter square. Moreover, upon performing the high throughput 16S rRNA gene sequencing of the anode biofilm. So we scraped the anode biofilm, extracted the metagenomic DNA, and then performed the sequencing and identified that the experimental anode system, that is CWMFC2 anode system, had an enrichment of 32.7% of the electrochemically, that is D-sulfo-bulbous, along with the other bacterial fiber. So D-sulfo-bulbous is also called a cable bacteria. Okay, it's also known as a cable bacteria. That means it produces nanowires for direct electron transfer to the anode. So that's why enrichment of this genera in the CWMC2 system can be the likely reason for higher power density outputs of the. So in addition to the treatment of the textile dye, real textile dye wastewater, we also we also wanted to study whether the system can also reduce the toxicity of the textile dye wastewater. So for that, we tested the toxicity of the treated and untreated wastewater on 9 tilapia animal models. That is Oreochromis niloticus. So as you can see, after 12 hours of exposure of 3 groups of fishes to the tap water control, untreated 30% volume for volume textile dye wastewater. So here we utilize 30% wastewater because if we use 100% wastewater, all fishes will die within 12 hours. Then we have the CWMFC2 treated wastewater, and we isolated the mRNA, and the mRNA was converted to cDNA, and qPCR was performed for hepatic oxidative stress biomarker genes like catalase, glutathione transferase, cytochrome P450, and superoxide dismutase. So, in all these biomarker oxidative stress biomarker genes, what we identified that untreated wastewater induced higher folds of expression, like 15 folds of expression to 3.5 fold expression to 2-fold expression to 1.5-fold expression. Whereas the CWMFC2 treated wastewater showed the gene expression of these hepatic oxidative stress biomarkers equivalent to that of the tap water control. So the treatment using this new technology, that is the CWMFC system, was not only effective in treatment of the wastewater, but it also effectively reduced the toxicity of the wastewater. So with this initial success of these lab scale systems, we further screen for various plants species to develop this system at the pilot scale. So up to 120 liters. So we wanted to demonstrate up to 120 liters what can be the treatment efficiency. So for that, initially we screen the plant species, various plant species. So for that, we screen various plant species like Primbistalis dichotoma, Elemus repens, Primbistalis ferrugina, and Typha augustifolia from various long-term polluted sites. Okay. So, Primbistalis dichotoma that was demonstrated in the lab scale system was from Maharashtra Industrial Development Corporation in Kolhapur, whereas the Primbistalis ferrugina and Elemus repens from Jaipur site near the Bhadar River. These plant species give maximum ADMI, that is American Dye Manufacturing Institute color index remote, as well as the COD reduction and maximum power density of 248.72 and 224.3 milliwatt per meter square. So based on these results, these plant species were selected to develop a pilot scale system. That is a two-step system, that is CWMLC1 as step 1 and CWMLC2 as step 2. And within this system, the step 1 system was planted with Firmicutalis ferrugina plants. And the step 2 system was planted with a plant-plant consortium having a Firmicutalis ferrugina and Elephas method. And the stainless steel grade 316 electrode were used for this experiment. So the filter media that was used was gravel and soil, and the effluent was inoculated in a stepwise increasing concentration from 200 to 1000 milligram per liter. And the treatment efficiency in terms of ADMI and COD removal was community DC5. And this DC5 community also generated 188.43 milliwatt per meter square of power density upon treatment of real textile dye wastewater. And this is the cyclic voltammetric analysis, which indicates, as you see, at 0 hour and at 96 hours. So this indicates the redox activity of the bacterial community. So that's why this bacterial community was used to bio-augment the two-step pilot scale system that was established. So the two-step CWMFC system was augmented with the enriched DC5 electroactive bacterial community at the CFU of 10⁷ CFU per mm. And before bio-augmentation and after bio-augmentation, the system was operated from 200 to 1000 milligram per liter of COD. And the system was also operated in the open circuit configuration and the closed circuit configuration. So in the NOC system, if you connect a resistor externally, that is called closed circuit configuration. And upon the continuous operation, what was found that after bio-augmentation, the treatment efficiency of COD removal improved from 62% to greater than 74%. Likewise, ADMI removal after bio-augmentation also improved from 90% to 97%. Mean percentage ADMI and COD removal showed an increase of 12% and 7.3% respectively after post bio-augmentation with the electroactive bacterial community EC5. Moreover, the power density increased from 177.3 milliwatt per meter square to 197.94 milliwatt per meter square. So a 20.64% rise in the power density was observed. Upon high throughput 16S rRNA gene sequence analysis, what we identified that the DC5 augmented bacterial genera were shown to persist in the CWMFC system. So in the HSCWMFC system, the bacterial genera like Firmicutes, Bacteriodota, and Proteobacteria were persistent in the augmented system, and these came from the DC5, the electroactive bacterial community that was bio-augmented. And within the genus, Exubacterium and Desulfovibrio and Bacillobacteroides were the most abundant genera at the anode surface of CWMFC1 and CWMFC2. And these genera are known to have members which are electroactive as well as dye-degrading. So, in a summary, to summarize the whole approach, so the textile dye polluted sites in Badar River in Jaipur, from where the two plant species were obtained, that is Pipistrellus ferruginea and Animus repens. And the rhizospheric bacterial consortia was enriched from the long-term polluted site of Kolhapur in Maharashtra. And a community of electroactive bacteria was enriched in a dual chamber MFC. And this enriched bacterial community DC5 was then augmented in a two-step horizontal subsurface flow constructed wetland microbial fuel cell. And upon bio-augmentation, it was found that the COD reduction of the system increased by 12%, whereas the maximum power density of the system was enhanced by 20.64 milliwatt per meter square. Likewise, the bacterial genera present in the electroactive community were also found to be enriched in the CWMFC system. So that is the up to pilot scale. Now the current ongoing research in our lab is focusing on scaling up this technology to TRR 5 level or 6 level, that is demonstrating it on site at Vatwa CETP. So we have our collaborator who is expert in the designing the electrodes or bioelectrochemical systems. That is Dr. Venkata Mohan from Indian Institute of Technology, Chemical Technology in Hyderabad, with whom we are designing flow-through wetland system. And through a circular bioeconomy approach, we are integrating the hybrid bioreactors with the flow-through wetland system to scale up the bioelectrochemical treatment technology. So in addition to bioelectrochemical treatments, we have developed several hybrid bioreactors for the treatment of real complex textile dye wastewater, which has a very high COD of 8,380 milligram per liter. So this wastewater was again collected from the local textile dyeing industry, textile dyeing industries from Vatwa, and this was composed of 27 different dyes, including azo and viscose dyes. So here again the same approach was followed. A bacterial community was enriched by more than 100 subcultures, and after high throughput sequencing, it was found that the enriched bacterial community or the consortia DR4 had presence of Comamonas genera in abundance. And upon sequencing and high throughput sequencing and prediction of the metagenome, predicted metagenome, xenobiotic degradation or metabolism genes were identified in the consortium. So this was the flask level experiment. And this consortium was then augmented into a hybrid fixed film bioreactor, which was again tested with various packing materials like wood charcoal, coconut coil, and PVC chips. And this bioreactor was compared. All these three systems were compared. And as it can be seen, the wood charcoal supports a massive biofilm growth for the consortium. And then it was chosen as a final optimized system, which was then run at different HRTs, like hydraulic retention. And it was found that 2-day HRT, the system gave better ADMI, that is color removal, 98% color removal, 90 and more than 98% COD removal, as well as more than 92% of COD removal at the optimized hydraulic retention time, along with the removal of other parameters. Then we scrape the biofilm from the electrode. So here first we followed the community dynamics on the upper zone, that is the oxic zone of the bioreactor, and the bottom zone, that is anoxic zone of the bioreactor, and identified what is the community dynamics across 5 different HRTs from 1D to 5D HRT. And what we identified that Enterococcus bacteroidetes, Pseudomonas, Bacillibacteroides, and Sulfurous bacteria. These genera persisted throughout the different HRTs. So that is composing of a core microbiome of the bioreactor system across 5 different HRTs. And the core microbiome of the bioreactor system shared several OTUs from 200 to 300 in the upper zone, oxic zones, and the bottom zone of the bioreactor. This bioreactor was then coupled with the aerobic system, and the aerobic bacterial community, sequential community, was dominated by Pseudomonas and Acinetobacter. So that is a hybrid system of which gives efficient treatment as well as the different bacterial genera that are responsible for this treatment were identified using high throughput metagenome sequence. Moreover, further, we also identified what are the genes that are encoded by these organisms. So for that, the short-term metagenomic sequencing of the biofilm was performed. And from this biofilm, we identified various genes responsible for, say, aminobenzoate degradation, benzoate degradation, various oxidoreductase genes within the xenobiotic metabolism. Novel oxidoreductase and azoreductase genes were identified from the bioreactor metagenome. And one of the genes we recently characterized as BM-AZOR, that is biofilm derived AZOR gene. So that was cloned, heterologously expressed in E. coli and purified. And its structure was also modeled. And the purified recombinant BM-AZOR gene was characterized. And it was found to be a thermostable FMN-dependent NADH azoreductase gene. As you can see from the phylogenetic tree, it clusters with the FMN-dependent NADH azoreductase gene. So it uses NADH as a co-substrate more preferably as compared to NADH. So this gene was also thermostable as predicted from computationally as well as through experimental validation. And it preferred methyl Z as a substrate. In presence of FMN and NADH, it gave 1.34 units per milligram of specific enzyme activity. So that is how you can identify novel genes from the bioremediation system through metagenome approaches. Okay, so with this, I would like to thank my group of students. Without their work, our research would not be possible. And the research presented today, out of the 11 students, is there of 2 of my students who are currently pursuing their postdocs abroad. That is Dr. Rohit Rathod and Dr. Disha Patel. So we are also thankful to the funding agencies who have funded our research very generously for around 3.7 crores of grants from 7 projects like GSBM, DBT, SERBM, and MC. So thank you for patiently listening. If you have any question, I will be happy to.
I have a question regarding the metabolic changes that also influence the
Phytoremediation.
not phytoremediation, phytoflora or whatever the plants that you have used. So, have you checked the any status of the plant metabolites?
So, our focus was mainly on the rhizospheric microorganisms,
Okay.
but not on the plant. So we haven't tested the plant enzymes or plant metabolites.
Okay. Any secretory products of the rhizomes?
Rhizospheric zones, plants are known to secrete root exudates,
Yeah.
which are rich in the carbohydrates.
So, is there any influence of the plant secretion on the?
Definitely.
Yeah.
Definitely, there would be an influence. So that's a nice question. Each plant will have a unique rhizospheric bacteria. So when you characterize using 16S rRNA gene, so it is not only the plant species, but the plant species associated rhizospheric bacteria that are also responsible for treatment.
Last question. So suppose if you have isolated some of the plant associated flora, how much chance there exists that when you apply the same for the large scale remediation of the soil, not only the water, but if you try the same with the soil, whether it will be effective or not?
That is what we isolated the rhizospheric consortium.
Yeah, but that is plant specific.
That is from a different plant. That
Yeah.
is the what we isolated is from Ipioma aquatica, and then applied it to Flimbristelis sparugina and Elimus repens. But still it worked, and it increased the system.
But
and
all these three are grass species.
Yeah.
So the scenario for the agricultural land would be little different for.
Yeah, definitely. Agricultural land, and in any case, you cannot use agricultural plant for phytoremediation. You have to use a weedy species or a non-medicinal or non-food crop variety. And all these are stringent weeds that will be able to sustain pollution found at long-term textile dye polluted sites.
Thank you.
Thank you.
So that was a very nice presentation. So I have a couple of questions. You mentioned NADP dependent azoreductase enzyme.
NADH, yes.
NADH, sorry. Sir, is the same gene responsible for nitrogen fixation as well?
No, azoreductase will be very specific for dye. For
Okay.
nitrogen fixation, you will have NIF genes.
Okay.
So azoreductase gene will do a reductive cleavage of
Right.
the azo bonds,
Right.
and it will not have anything to do with the
With
nitrogen.
the nitrogen fixation. Okay. So second thing, is this microbial fuel cell completely anaerobic?
No, the anode is anaerobic as I showed, and
Okay.
the cathode is aerobic. So cathode you put an aspirator and put oxygen, so
Okay.
that cathode can have the oxygen which will act as an electron acceptor.
Okay, like the one that we have in aquarium, like
Yeah, same aspirator on the lab scale. Yeah.
on the, okay. Fine. And so the third question, the amount of energy that you are getting, does it have any correlation with the composition of the affluent?
It does actually. That's a very nice question. So if we see the textile dye wastewater generates more power density as compared to other types of wastewater because textile dye itself acts as a redox mediator. So it itself acts as a, or its intermediates that are present in the wastewater. They act as redox mediator. Whereas other wastewater may not give that much output, power density output. So it will be influenced by the wastewater. It will be influenced by the bacterial community, the electroactive bacterial community that is used.
Sir, have you tried this the same concept with fungus?
We haven't tried with fungus. We have just focused on the bacterial systems. And even with the plant systems, we have just used the rhizospheric consortia and not the plant system as a whole.
Thank you, sir.
Sir, I have a question. Sir, I think you have shown two different sites. That is one Karnataka and one is Jaipur. If I'm
One is Tirupur in Tamil Nadu
yeah,
and one is in Jaipur,
Yeah.
which you might be very familiar.
Yeah.
And another was Sabarmati.
yeah. So consortia of one plant from one site has been used for the other one.
For augmentation in the other.
So why can't we use the same Jaipur site?
Yeah, we use the same also. So I showed you that we compared 7. And from the 7, the one which was the best electrochemically active consortia, that we used for augmentation. Yes, yes.
Cell dye. There's some dye are degrading by bacteria. So how can identify the degradation pathway with the help of
Yes. So can you guess any analytical technique through which you can identify the degradation pathway?
CMS?
LCMS or GCMS. So LCMS is high resolution LCMS that we have. So in that we identify what are the intermediates found. Mostly the first intermediate is aromatic amine. And then you want that aromatic amine to be degraded to or completely mineralized because aromatic amine would be much more toxic as compared to the parent compound. So you study the intermediates, mostly the gentisate pathway or the benzoic pathways are what they will follow upon degradation.
So when we took sample from the river side and so there is a sample is going to with a mixture of dyes. So
Yes.
K2 dyes and nitrogen based dyes, sulfur dyes.
Yeah, it can be, say if you are going for wastewater, as we went for wastewater, it can be polluted with multiple types of dye. That one study that we showed, 27 different dyes were there.
Specific bacterial species degrade specific dye.
That is for mechanistic study. If your aim is for mechanistic study, then you can get the specific dye. Say industries will give you the pure compounds and then focus on the fundamental research. So what we were focused on the applied aspect, that directly find a solution for treatment of the wastewater because normally the bacteria are also very specific for the structure of dye. So one strain can degrade a particular dye, but other cannot. So that's why it's better that you use a community which is already acclimatized in that wastewater for several subcultures. So there are more than 100 subcultures. You have been enriching that community. And all the community members are capable of degrading the complex dye mixtures.
So, sir, take the top. Yes, sir. Hello, I just placed you phone. I would like to request Dr. Guru Pandit, HOD Microbiology Department, to offer the token of love from Atmiya University. So please. And the felicitation certificate from the cert. Thank you so much, sir. We will discuss for 15 minutes for the tea break. I would expect after tea break, we'll all meet again for a very literary session. Thank you all. Request all to please take the seats. Maintain the ambience of the auditorium, please. Please be seated. We are about to welcome Vice Chancellor, Atmiya University, Dr. Shree Tripathi, sir. We welcome you, sir, to the valedictory session of National Conference on Environmental Microbiology and Regulatory Aspect, which is jointly organized by Department of Microbiology, Atmiya University, and Shree Virani Science College, Rajkot. The national conference has been supported by DBT, CSIR, Government of India. I would like the dignitaries to take their seats onto the dais. I would request VC sir, Professor Shree Tripathi sir, Dr. Chirayu Desai sir, Dr. Neepa Pandey, Dr. Rohan Pandya sir to take their seats onto the dais. On duly welcome Dr. Ashish Kothari sir, Director, Research Innovation and Translation, to join us directly onto the dais, please. I will also take the privilege to invite organizing secretaries, Dr. Abhijit Joshi and Dr. Raksha Bavankar Madam to join us on this. The organizing committee was headed by Visu. A big round of applause. And Virani, I would request to welcome every dignitary here. Request Dr. Rohan Pandya to present the floral welcome for VC sir. I would request Dr. Neepa Pandey Madam to florally welcome Chirayu Desai sir. I would request Dr. Abhijit Joshi sir to welcome Kothari sir. Dr. Raksha Bavankar Madam to welcome Neepa Pandey Madam. Dr. Bhargav Vaghela sir to welcome Rohan Pandya sir. Now, I would like to felicitate the conveyor, both the conveners and the organizing secretaries of the national conference. I would request Vice Chancellor Sir, Dr. Shree Tripathi, to present the felicitation certificate to both the conveyors, Dr. Rohan Pandya and Dr. Neepa Pandey. I would request Dr. Ashish Kothari sir to felicitate the organizing secretaries, Dr. Abhijit Joshi, Dr. Raksha Bavankar. Let us all be fortunate enough to listen to Vice Chancellor Sir's special address to the session. Please, sir.
Thank you very much. Very good evening and namaskar to all of you. Dignitaries on the dais, off the dais, Dr. Desai, Dr. Pandey, my colleagues, faculty members and students. First, I am so happy to see the level of energy during the valedictory program. This itself shows that what the journey has been for the last 2 days. On behalf of our mentor, our president, Param Pujya Tyag Vallabh Swami, our honorable Pro-Chancellor, Professor Dr. Sheila Ramchandran, and on my own behalf, I would like to first congratulate the organizing secretaries who led this conference to make it a successful story. Congratulations, Dr. Joshi. Congratulations, Dr. Bhatt. The force behind them is the one who is leading and supported by the convener from MNN Virani Science College. So Dr. Pandey, Dr. Pandey, congratulations for you. And the second level of leadership is someone who is heading the Directorate of Research, Innovation and Translation, my colleague Dr. Kothari, and who is at the foundation, who is inspiring all the research activities happening. So congratulations to you. RIT is there in the logo as one of the co-partners. A big thank you to MNN Virani Science College. And definitely although I was not present yesterday, I missed it to be with you all during inauguration. But I was going through the proceedings of yesterday and I was looking at such an impressive line of speakers who joined yesterday. A lot of efforts happened. I am not from the microbiology subject. I don't have much of understanding of how microorganisms function and how it is researched. But just my opinion on the topic, because this is so much interdisciplinary or transdisciplinary in nature, because we are talking about regulatory aspects of microbiology. One thing what I understand, today you all know, and I will not be talking on this from expert perspective as a microbiologist. But what I would suggest that in our day-to-day lives, in most of the areas where you see that it has affected from healthcare to nutrition to hygiene to pollution, and everything is now microbiology has its application. But when it comes to make it a practical application which can be accepted at the level of individual, families, societies, in our societies, it has to go various tests, what we call economic feasibility test. It has to go social feasibility test, and there comes the role of regulations. So I'm sure that based on the last 2 days of discussions, you would have developed good insights into what are the different angles of regulations. It is not just one regulation or law. In light of that, we are developing the application. But we have to think that how this innovation is going to align and address the issues within the framework. And sometimes if frameworks required to be changed, it is the responsibility of the researchers to inform the policymakers that what type of the changes should be brought in the regulatory aspects as well. So those of you, I'm sure many of you would be having interest in this area, and I'm sure in future try to look into, you try to extend this conference into application, how you can find something, how you can work on this interdisciplinary area of this innovations of microbiology, interacting with the laws and regulatory systems, and what few things we can do then you will be making it a successful and impactful. My friends, I'm so happy to be here with you. Congratulations to all of you once again. Thank you so much for your time.
So now is the time to distribute the prizes. Finally. So we have two line of presentations, oral presentations and poster presentations. Both of them were conducted in both the hybrid ways, online oral presentation, offline oral presentation, online poster presentation, and offline poster presentation. I would start with the third winner, followed by second and then the third, then the first, sorry. I would start with online oral winners, right? The third prize goes to Dr. Rugesh H. Trivedi and Ms. Hemlata Sharma. The second prize goes to Dr. Vishali Bagh from Naonal College of Professional and Applied Sciences, Navalben. And the first prize goes to Ms. Aarti Ponraj from B.S. Abdul Rahman Crescent Institute of Science and Technology. The second pack of online poster winners are, the third prize goes to Bala Murugan from IARI, Indian Agriculture Research Institute, Pusa, New Delhi. The second prize goes to Gamit Amit Kumar Bagirish Bhai from CG Gupta Institute of Biotechnology, Uka Tarsania University. And the first online poster award goes to Kavya T from the Indian Agricultural Research Institute. Congratulations to all the winners. So now is the time for offline oral winners. The third prize goes to Kacharia Dimple from Atmiya University. And I would like to request, I'm so sorry, I just got driven away. It is my pleasure to see the award. So we would probably vote, it's a tie. And the second member is Janvi Parnal. Please come up on stage, Janvi. She belongs to Province India. The third prize for offline oral presentation is Janvi Parnal. Thank you, sir. The second prize, I would like Dr. Chirayo Desai, sir, to give away the prize. The second rank goes to Hetvi Wang, again to my favorite. I would request BC sir to please present the first prize for oral presentation. The first prize belongs to Kunal Kumar Dhulia from the Arts. Thank you, sir. Now, offline poster winners. I would request again Dr. Ashish Kothari sir to give away the third prize. The third prize goes to Sunnati Yagnik. Again, to Matthew. And sir, I would request Dr. Chirayu Desai sir to give me the second prize, please. The second goes to Mr. Yash Kumar Patel from PD Patel. Sir. Sir, I would request Dr. Shiv Kumar Tripathi sir to give away the first prize. The first prize for the offline oral presentation are, poster presentation are Ms. Riti Anand and Mr. Rohit Kumar Mahto from Usha Martin University, Ranchi. Thank you so much, sir. Now, I would like to hand over the mic for the conference summary by Vijay Joshi, sir, so as to enlighten us at what is the conference summary.
Thank you, Shweta, to call me on the steps. Just by chanting the name of Lord Swaminarayan, his disciple, Brahmaswarup Param Pujya Hariprasad Swamiji, Pragat Guru Hari Swarup Swamiji, and our mentor, Param Pujya Tyagulab Swamiji. Honorable Vice, sorry, VC sir, it's my privilege to give you a summary of this conference. The journey of this conference is actually starts from the April month, when we conducted a meeting to propose the account or rather than academic planning. And in that, we just proposed that we will plan a national conference in the month of February. On the 3rd of August, we took the decision and we finalized the theme. We also approached our respected leadership team, and we also got their approval. Yes, you can proceed for the same. Later on, we find out the respected speakers among the various institutes of India. Our target is not to just call scientists or academicians. That's why we decided that one group should be from the industry also. So we approached, and Dr. Manish Sir accepted our invitation, and he today delivered his talk on the same. After finalizing, we approached towards the three funding agencies, and our success rate was just this time is 66.66% because of we got the approval from the only two, not from the three one. Next time, definitely, sir, we will try to get the third one also. After that approval, we start our planning, and as we open the sessions, or we start circulating our brochures with all over the India, we got a huge response, but unfortunately, we run down only at 249. So we lost one just wicket, and we required one more if it is 250. So the summary is like this. Only 249 participants are currently present, or they enrolled themselves for this national conference. Among them, 120 are in online mode and 129 is in offline mode. Another thing is that mostly research, this time we found that research scholars, 42% of research scholars are present in this hall, and also they are listening to us through this live tube. I'm happy to share also sir, that among that 249, 40% means 61 participants present their research work either in the oral form and in the postal form. Totally, there are 24 number of research scholars who presented their work, and remaining are the different academic backgrounds, means 37 they are from different academic backgrounds, including the researchers, including the academicians, and the result already ma'am declared to you all. So it is known. I'm also present here to summarize the things. We found some hurdles here to organize the things, but it gives boost to all of us that next time also we can plan something, and yes, by the team we are able to achieve that also. With this, my words, I conclude my session. Thank you so much for listening.
Thank you, student sir. I would like to invite Dr. Raksha Pavankar Madam for the vote of thanks.
Namaste, who are not present here. Jai Swaminarayan. It is with immense gratitude and sense of accomplishment that I stand before you today, representing the organizing committee of the National Conference on Environmental Microbiology and Regulatory Aspects. It was jointly organized by Microbiology Department of Atmiya University and Shree Virani Science College, Rajkot. I extend a heartfelt thanks to each and every one of you who contributed towards the success of this conference. First and foremost, I would like to express our deepest gratitude to Brahma Swarup Hari Prasad Swamiji, Pragat Guru Hari Prem Swarup Swamiji, and our mentor Param Pujya Tyagala Swamiji Maharaj for their blessings and unwavering support throughout this journey of the conference. We are profoundly thankful to the management of Atmiya University and Shree Virani Science College for their continuous support towards the smooth conduction of this national conference. My special thanks to the leadership team, respected Pro-Chancellor Ma'am, Dr. Sheila Ramchandran Ma'am, respected Vice Chancellor, Professor Shri Tripathi Sir, respected Pro-Vice Chancellor, Professor Jayesh Deshkar Sir, respected Dr. K.D. Ladwa Sir, Principal of Virani Science College, Rajkot, respected Registrar Sir, Dr. D.D. Vyas Sir, respected Director, IIT, Dr. Ashish Kothari Sir, Dr. Rohan Pandya Sir, HOD of Microbiology Department, Atmiya University, Dr. Nipa Pandi Ma'am, HOD of Microbiology Department, Shree Virani Science College, Rajkot, for their continuous support and guidance during the preparation and execution of this conference. Thank you. I extend my sincere thanks to all the deans of the Atmiya University and all the head of departments of Atmiya University and Shree Virani Science College for their continuous support towards the success of this conference. We are immensely grateful to the Department of Biotechnology DVT and CSIR Council of Scientific and Industrial Research Government of India for their generous financial support which made this conference possible. I extend my sincere gratitude to all the respected speakers, Professor Hemant Purohit Sir, Professor Sasikala Ma'am, Professor Dayanand Aggar Sir, Professor Arun Kharat Sir, Dr. Madhvi Joshi Ma'am, Dr. Chirai Desai Joshi Sir, Dr. J.N. Joshi Sir, Dr. Manish Raj Sir, and all the respected judges and all the participants from all the 14 states of India in the online as well as the offline mode. A special mention goes out all the committees who worked towards the tirelessly behind the scene to ensure the smooth functioning of this conference with every aspect of this conference, and this belongs to stage committee, registration committee, food committee, photography and videography team, accommodation team, transportation team, discipline committee, designing and printing team. We deeply appreciate the dedication and hard work of all the teaching, non-teaching staff, research scholars, all the student volunteers, all the peon guys,
and who contributed towards their time and effort to make this conference a resounding success. Together, we have taken a step ahead, forwarding in advancing understanding towards the environmental microbiology and the regulatory aspects. So, once again, I deeply sincerely thanks to each and everyone. Thank you so much. Yes, Swami.
So much, ma'am. Just, all to just gather upstairs for the group photo. I would request all these committee members and the team to come up on stage for the group photo, please. Please join us on stage, everyone. Committee members, please. The dignitaries can sit on the dais. We'll arrange ourselves behind them. I would request the research scholars, everyone involved in helping the success of this conference, everyone should join on stage. Everyone.
Yes.
Anyone, if anyone is voluntarily wanting to give the feedback on the national conference organized. Anyone like to give the feedback? voluntarily? Please raise up your hand. Find it. Let us declare the wrap-up of National Conference on Environmental Microbiology and Regulatory Aspects closed. I would request all to stand on the seats and join us for national anthem. I would request the HOD sir to escort the dignitaries of the dais. Please stay seated till the dignitaries leave the auditorium, please. Stay seated, everybody. There's a little announcement for you guys. Those who wants to collect the certificates. But then. Thanks.